Hi, today we'll be talking about the activation of T cells.
So what are T cells?
T cells are a type of white blood cell that are of key importance to the immune system and a part of the adaptive immunity in which there are two responses: humoral immunity mediated by B lymphocytes and cell-mediated immunity mediated by T lymphocytes, which we will be talking about. Adaptive immunity is a system that tailors the body's immune response to specific pathogens. The T cells are like soldiers who search out and destroy the targeted invaders. They originate from the bone marrow and mature in the thymus.
So what is T cell activation?
T cell activation begins with T cells entering the thymus where they proliferate. These two subsets T cells are called CD4 positive and CD8 positive. CD4 are helper T cells and CD8 are cytotoxic that will kill infected cells. T cell activation is a cell-mediated response occurring in three simple stages or signals.
Signal 1 causes activation.
This cell involves antigen recognition, which is the regulated movement of a signalling receptors and adhesion molecules at the immune synapse.
So to explain this further,
There are three types of antigen-presenting cells: dendritic cells, macrophages and B cells. Let's use dendritic cells such as Langerhans as an example. These take up and process the antigen in the epidermis or the oral epithelium.
The process starts as dendritic cells phagocytose the antigen, degrading it into proteins. These fragments of proteins are presented onto the cell surface via MHC class II receptors, which are receptors found on antigen-presenting cells. So basically, MHC class II receptors is how the antigen fragments are mounted onto the cell surface.
After this, the antigen is now bound to the MHC class II receptor, which is present to the T cell using a T cell receptor, which is simply a receptor present on the surface of T cells. There is a huge availability of T cell receptors as different receptors bind to different antigens.
the macrophage will move around until it finds the correct T cell with the correct T cell receptor. This is a specific response. Therefore, you need the TCR complementary to antigen you're presenting. More specifically, the MHC class II binds with the CD4 cell receptor on the T cell, which provides immune protection, and MHC class I binds to CD8.
Note that the activation of T cells may require multiple or prolonged TCR antigen interactions.
Signal 2 involves co-stimulators.
This phase ensures that the immune system responds to microbes and not to harmless antigenic substances. Second signals for T cells are co-stimulators on antigen-presenting cells and cytokines produced by antigen-presenting cells.
The concept of co-stimulation is consistent with the characteristics of a newly identified molecule called CD28, which expresses on naive CD4 and CD8 positive T lymphocytes. An important example of such signaling at this stage is CD28 binding to CD80 or CD86, which ultimately signals for the survival and clonal expansion of T cells.
Signal 1 but no signal 2 leads to anergy, which is a tolerance mechanism in which the lymphocyte is functionally inactivated following an antigen encounter, but remains alive for an extended period of time in a hyporesponsive state. Signal 1 and 2 together drive the cell into signal 3. This is the final signal.
Signal 3 involves cytokines which are produced by APCs or T cells.
It is the cytokines which stimulate T cell expansion and differentiation into effector cells and hence determine the subset of T cell differentiation. Once activated, T cells leave the tissues and migrate through lymphatics back to the bloodstream.
Most T cells undergo expansion and migrate to the periphery to become effector cells as they lack CCR7, which is chemokine receptor 7. Some express CCR7 and stay in the lymph nodes as central memory cells.
So what's the dental relevance of this?
Periodontal disease is a peripheral infection involving species of gram-negative organisms characterized by necrosis of gingival tissues, the periodontal ligament and alveolar bone. T lymphocytes can be found in a dense inflammatory infiltrate in this disease. CD4 positive and CD8 positive T cells are present in periodontal lesions, as are memory T lymphocytes.
Another disease is oral lichen planus, which is a T cell-mediated autoimmune disease. It's a chronic inflammatory condition that affects the mucosa membranes inside the mouth. It may appear as white lacy patches, red swollen tissues or open sores. These lesions may cause burning, pain or discomfort.
Thank you for listening, and we hope you've learned all about T cell activation.